LCC, and welcome to September's version of the monthly webinar. Today, we will be hearing about the Canadian River Project, which was funded through the South Central Climate Science Centre. This project evaluates long-term trends in stream flow and surface water quality and aquatic ecology to project changes in those metrics that could occur given various climate change scenarios. We've got two presenters today. Bill Andrews is director of the USGS Oklahoma Water Science Center and has worked for the USGS since 1988 in Florida, Minnesota, and Oklahoma on a variety of topics. Bill has a bachelor's from the College of William and Mary in geology, a master's from Northern Illinois in geology, and a PhD from the University of Oklahoma in environmental science. So lots of good experience there. Bill is a longtime professor, or was assisted in this project by Dr. Baxter Vio, a longtime professor of water resources at the School of Civil Engineering and Environmental Science at the University of Oklahoma. And then our second presenter today is Dr. Thomas Worthington. Tom was a postdoc researcher at the Oklahoma Cooperative Fish and Wildlife Research Unit at Oklahoma State at the outset of this project, and is continuing to assist on the project as a research associate at the School of Biosciences at Cardiff University in Cardiff, Wales. So he's joining us from across the pond today. So I think we're ready to get started. I will turn it over to you, Bill.
Hi, Cynthia. Thanks for the introduction. We appreciate being invited to tell all of you about our project, give you some highlights of the project. I'm just going to jump in from slide number 1. Let's see. As you said, it's, hang on just a second here. Okay. Sorry, I had a little control problem there. The project history is that we started right about 2 years ago. We have a 2-year duration. As with many complex projects, we're running a little late. I'll discuss a few of the reasons for that. But the primary objectives of the project are to investigate the effects of climate change on stream flow, water quality, and fish in the Arkansas Red River Basin. And I'll discuss a little bit more about how we narrowed down to the Canadian River Basin. So there's really kind of two parts. One is looking at past trends and also projecting. I think Baxter View is going to be able to join us for several of these slides. Baxter, if you're on, if you can do, what was it, a *6, Cynthia?
Yes, to unmute.
To unmute, so hopefully he'll be able to join us. And the main project goal, the real end product of this, is to provide information that's needed and useful to land managers and others to ensure the sustainability of aquatic wildlife with changing climate conditions, with some of the projected changes in stream flow and water quality that we've seen. Okay, so the first part is kind of a list of issues with starting the project. I'm sure many of you have had complex multiplayer or multi-participant projects, and sometimes they take a little while to get off the ground, so I'm not really going to get into the details. But as is common for these projects, they do tend to go a little bit longer than originally planned. Okay, getting into the project progress. I've been the lead of the project since November 2013 here at the Oklahoma Water Science Center. My main component was a water quality trend analysis that I completed in early spring of 2014. I also did some work on the data management plan and loading some of our data and metadata into ScienceSpace. For anybody in USGS, we're all going to be doing a lot more of that in the coming years. We have had monthly conference calls since we were scattered around the country, and now we're actually scattered on 2 continents now. And we've also included members, particularly in the last 5 months or so, from the 2 major LCCs interested in this basin, the Climate Science Center who funded it, and representatives of a sister project down on the Red River being done by the Chickasaw Nation and some of their partners. Okay, we have given a couple of talks at the American Fisheries Society annual meeting last year in Quebec City, this year in Portland. If anybody wants more details about that, I can send out copies of those PowerPoints. Basically, the water quality and the work that I did and the rainfall runoff work of the project that Baxter View oversaw and finished up are pretty well wrapped up. The analysis of ecological data and ecological flow determinations are ongoing. And in addition to the talks, we plan on writing between 2 and 4 journal papers, and there's also discussions about an interactive website, which Tom will talk a little bit more about that later. So like any project like this, we do want to have, basically have the results be useful to other people outside of the project, and to other, or basically to land managers, citizens that are interested in water resources in this area. So a large part of that outreach to the public and land use managers will be a web-based decision support tool. And Tom is really the engine behind that getting done, and we'll be working with him on that over the next several months. So let's go ahead and take a look at a few slides of results. The first part here, the first couple of slides relate to my work. So what we did was look at trends and concentrations of selected water quality constituents, relatively simple things, physical parameters like temperature, pH, concentrations of chloride, suspended sediments, and dissolved oxygen. Working with ecological members of the team, they thought these might be the most important water quality constituents that could affect fish communities in the river and its tributaries. So we were looking for sites with long-term streamflow records, so streamflow gauging stations operated by the USGS, and also sites that had substantial water quality data, hopefully continuous data for several decades, but at some sites they were kind of discontinuous with several-year breaks between data collection at the site. So we sampled both on some tributaries and the main stem of the Canadian River. The source of the water quality data were US EPA STORAT and USGS NWIS. The streamflow data came from USGS NWIS, and there were some streamflow data in STORAT. When we had sufficient numbers of contiguous water quality data, we did use the LODES program to estimate mean annual flow-weighted constituent concentrations for dissolved oxygen, suspended sediment, and chloride, not really the water temperature and the pH for those. We use mean annual values of the data that we were able to get. Kendall's tau was used either with flow-weighted concentrations or the mean annual values versus time to try to look at the presence of significant trends or absence of such trends. So just one simple slide on water quality trends. We did do some linear regressions with streamflow, and those indicated generally negative relations between streamflow and chloride. So essentially the chloride's coming in largely in the base flow components seeping to the streams from groundwater. Also negative relations between streamflow and DO and pH. So in the case of DO, it may be organic matter coming into the streams through overland flow during wetter periods. We did, as you might expect, find positive relations between stream flow and suspended sediment. That's quite common basically everywhere you look. The little graph up on the upper right is looking at mean annual flow in CFS near the state border between Texas and Oklahoma. So the bottom line is the mean annual flow in cubic feet per second. You can see, though I didn't fit a low S line or straight line to that, you can see a slight downward trend. But the red dots being the flow-weighted concentrations of chloride, the annual flow-weighted concentrations, you can see those generally going up during the 70s and 80s, and then kind of leveling off after the early 1980s. So it in large part does follow in a negative correlation sense with the stream flow at that site. And the long-term positive trends with time were most common for chloride and water temperature at these sites. And in terms of negative correlations, there tended to be decreases in stream flow. So the next area we were going to get into is looking at hydrologic effects projections. This part was done largely by Baxter View with some help by a USGS employee named Sam Rendon. Baxter View used the VFlow program and used radar-based estimates of precipitation in the basin from 1995 to 2014. Climate change projections were done with the downscaled IN-MC3 version of the CMIP3 model. Has Baxter been able to join us yet? Baxter, if you're there, can you hit *6? Okay. I guess Baxter wasn't able to join us, so I'm just going to have to sub for him.
Okay. I joined.
Oh, terrific, Baxter. Can you see the slides?
Yes, I do. Okay.
I'm on your second slide if you want to jump in and offer some more explanation.
Well, you're doing fine. I hesitate to interrupt.
Oh, no. You know more about this component.
Right. So we calibrated the VFlow model using historic rainfall, which consists of radar and rain gauge information. You can see the distribution of rainfall on the left with the kind of red blotches are the highest rainfall, and this is a total over a certain period of time. But what is important there is the variability of rainfall over the basin. And so we calibrated the model. You can see on the right the observed and simulated results, observed being the red dashed line. And you can see relatively good agreement between the peaks of the hydrographs and the low flow and so forth. And that was for about a 15-year period starting in 1994 going forward to 2014. That calibrated model then is used with perturbed inputs that represent future climate scenarios. So next slide.
Okay. Okay.
Yeah, so this, before we go on, that happened to be at Calvin, which is just upstream of Lake Eufaula. Okay. So this USGS gauging station. So now for the perturbation, what did we do? Well, altered two different components of future climate, and I'll tell a little bit about how we did that. But basically, we changed the precipitation and perturbed the historic rainfall based on what the ratio of a climate model would say the latter 30 years of the 21st century compared to or as a ratio compared to the latter 30 years of the 20th century. And so basically we see with this particular climate model, the INMC3 was chosen because it was represented 90th percent, or the reduction in precipitation, the 90th percentile, and an increase in warming at the 90th percentile. So warmer and drier, and so you can choose, almost any climate model and get almost any result. You can get status quo, no change at all, or it could be wetter, or it could be drier, and so forth. And so this is what was selected, and to investigate what would happen under warmer and drier conditions. And you can see climatology of discharge expressed as a monthly average. So the perturbed and baseline are shown on the left, and the discharges in CFS. And so, for example, a 200 CFS discharge would, on any one of those lines would represent the average flow for a given month. And so you can see, as expected, an increase in the spring period, but the baseline is quite a bit higher than the perturbed, and this is under future precipitation, but using historic PET. So we left the PET, which is the potential evapotranspiration, which is most directly related to temperature, and we left that the same and just perturbed the precipitation, and you can see a reduced runoff. And again, that's another USGS gauging station, but this time it's the Canadian River near Canadian, Texas, which is quite a bit upstream near in the Panhandle. Okay, next slide. Okay, so you can see a decrease. That's what the arrow is showing. So increased but now you see the next perturbation, where we leave the precipitation the same as the historic period, but alter the evapotranspiration due to warmer temperature, and we see very little effect. You can see that the baseline and perturbed lines are very similar, and again, that's at Canadian, near Canadian, at a USGS gauging station. And basically what that's telling us is that runoff is limited by the available precipitation, and you can't get drier than dry, as I am fond of saying. And so in these types of watersheds in this particular area, I think the fact is that there's plenty of potential evapotranspiration, or PET, but not plenty of precipitation. And so whether you get runoff or not largely depends on the availability of precipitation. Next slide. And this is just showing this in a little different way as a cumulative runoff plot, and its depth in inches expressed as a watershed depth. So if we took all the volume that ran off in a given year, this is again back down right above Lake Eufaula, you can see the baseline and perturbed, where this is under the assumption of future precipitation and PET, so both, although it'd be very similar to what you get with just perturbed precipitation. And so if we take that volume that runs past that particular stream gauge and average that over the watershed, this is a kind of a typical way of interpreting runoff volume. And so it makes it very intuitive compared to precipitation depth. And so you can see with the baseline that over this 15-year period, you get about 15 inches of runoff, but with the perturbed data set, you're going to get maybe 10, 11 inches of runoff. And you can see it following a similar kind of trend, but that's just the cumulative runoff. And that's actually been summed up on an hourly basis for that entire 15-year period, because the model runs with rainfall inputs that are at 1 hour time increment. Okay, next slide if there is one. Yeah, so you can see a decrease there. Yeah.
Okay, yeah, thanks Baxter. Those are great explanations. I think what we're going to get into now is switching over to Tom Worthington. I can say a few things about this particular slide and then jump over to my colleague Tom. Essentially, this is a schematic diagram of the structure of the project. The part that Baxter was just talking about is kind of the upper left-hand boxes with the high-resolution radar rainfall data, kind of the V-flow, and what we don't show is kind of plugging in climate change model. Then over on the lower right-hand side, we get the work that I talked about a few slides ago with collective parameters and load est and trend analysis. Now what we're going to talk about is the ecological component. There's kind of those bottom three boxes and circles down on the lower left. With that, I'm going to jump into the next slide and go over to my colleague Tom Worthington.
Okay, well, good afternoon, everybody. As I said, my name's Tom Worthington, and I was a postdoctoral researcher in Oklahoma. And kind of the main area that I was looking at was the ecological side, along with my supervisor, Dr. Shannon Brewer. And one of the questions that was brought up is, so why did we choose the Canadian River? Well, there are a number of very interesting ecologically speaking species within the river. So, and this is linked into some previous work we've done from the LCCs, looking at the Arkansas River shiner, one of the pelagic broadcast spawning cyprinids. And a number of these species are of conservation concern. And we've seen kind of a change in the community composition through time. And the idea was to look at how that was related to the flow discharge relationship that Baxter and had created using the B-flow model. One of the things that we wanted to do was slightly move away from looking at taxonomic distributions of species and look at species traits to kind of describe the whole community. And also kind of look at community change in relation to the flow regime. So next slide. So as a bit of a background, we're looking at the Canadian River Basin, but we're looking at a specific portion. So we have from Ute Lake in New Mexico down to Lake Eufaula in Oklahoma, with obviously Lake Meredith kind of splitting this up into two main fragments over in Texas. This area is known for being one of the remaining areas where some of these species, including the Arkansas River shiner, are still found, and therefore it makes kind of an interesting ecological case study. Next slide, please. So one of the first steps of the project was to translate the flow regime that's been created by Baxter and his team from the B-flow model and convert that into ecologically relevant flow metrics. And obviously I won't go into all of them because there's 181 different ones, but they relate to a number of different groups. So the magnitude, including kind of the average, low and high flows, the duration of flows, timing, frequency, and rate of change. So we've got a number of different variables which we could then relate to the ecology. Next slide, please. And the ecology in this case was previously collected fish community data from around 75 sites throughout our study basin. As you can see, they're kind of quite well distributed, particularly within Oklahoma. They're a bit more clumped in Texas and New Mexico. And this was data we gathered from a number of 8 state agencies, also U.S. Fish and Wildlife Service, and also some data from the published literature to kind of build together a picture of the communities within the Canadian River main stem itself and also some of its larger tributaries. Next slide, please. And as I mentioned earlier, we translate this sort of taxonomic identity into species traits. And this has the advantage that we can understand adaptations, maybe causal mechanisms for the relationship between flow and certain trait groups. It also allows this work to be interesting more than just the Canadian River and kind of be applicable across the US and look at relationships in other rivers that have undergone similar changes in discharge. And it also allows you to have a better insight into, say, rare and invasive species. Next slide, please. So the traits that we looked at, they were in 2 main groups. First of all, based on measurements of the specimens that were captured, that were carried out by PhD student Bob Mollenhauer at Oklahoma State, and looking at a number of factors that would influence the fish's behavioral and ecological responses. But one thing that's been highlighted with these species is there's been a change in the composition, as I highlighted earlier, particularly a change related to this one group, the Pelagic Broadcast spawners. So we wanted to have a look at the reproductive groups that make up the communities that we were seeing. So we looked at some broad reproductive guilds and also some much more specific guilds, and used that to split the community data into certain groups. Okay, next slide, please. The analysis that we're going to present now is using a fairly new package within the R software called MVAbund. And it's been designed to analyze multivariate data but move away from some of the sort of classical tests and fit general linear and linear models to each of the response variables. And in our case, the response variables are the different trait groups that I'm going to highlight in a minute. These could be those trait groups, or they could be individual species. The idea behind it, and this was pioneered by someone in Australia, a guy called David Wharton, is that it allows greater interpretability of the relationship between individual portions of the community and the drivers, which in this case are the flow regime. And from a kind of statistical point of view, it has the advantage of allowing kind of easier assumption checking to see how well these models are fitting. Next slide, please. So, as I mentioned earlier, we started off with a potential number of ecological relevant flow metrics. And as you can see, we have a large number in certain groups, lots of ones looking at magnitude, less looking at, say, rate of change. Obviously, we couldn't relate the community data at 75 sites to 181 different metrics. That's just statistically redundant. However, we came upon a method for reducing the number of flow metrics to start with. So can you press forward? First of all, this was based on correlation, so we tried to remove really highly correlated flow metrics as they become redundant in a statistical model. And we also used expert judgment. So we're looking for particular flow metrics that we thought were important to the species we were interested in. And to try and keep at least one in each of those categories, so at least one in each magnitude group, so one average, one low, one high. And we ended up with a subset of about 20 metrics. Next slide, please. And this is what we ended up with. However, that's still too many for the analysis that we wanted to undertake. So next bit, please. So again, we then reduced that subset using a statistical process called a stepwise variance inflation factor. And that's looking at how the variables interact within a model and maybe are explaining certain portions of the variability, and then to try and remove those ones that aren't contributing new information to the model. Next slide, please. And so this is the final candidate set of 8 flow metrics. We have a couple that look at magnitude, both average and high flows, some looking at high and low durations, flood frequency in terms of the frequency variables, and a number looking at timing, both predictability and the date during the year of the annual maximums and minimums. The rate of change variable actually dropped out once we used this variable inflation factor. Next slide. We then split the fish community. So this is the list of species that we collected from the data, and we split that up into what we consider kind of ecologically relevant groups using hierarchical clustering. The number of groups was split based on looking at the data and thinking how many groups seem sensible, but also based on some initial exploratory analysis which suggested, based on bootstrapping, that there was roughly 9 groups within our community. Next slide. And these groups kind of fall into 3 main categories. We have some that are very much driven by their morphology. So, for instance, the gar species are obviously very different morphologically. They have slightly different reproductive strategies, but they're still very closely grouped together. Some species are, on the other hand, driven by a mixture of morphology and also reproduction, such as the catfish and the bullhead species. And then we've got some reproductive groups, and the one to the far right in the light blue is the Pelagic Broadcast spawners. And the interesting thing here, it's moved some of the species from what would be essentially just morphologically driven, so having all the chub species together, and all the minnow species together, and all the shiner species, and it's split them into this mixture of morphology, so they're all cyprinids, but they're also have the same reproductive strategy. Another interesting point is chub shiner, which we don't know that much about, actually is believed to be one of these Pelagic Broadcast spawners, and it fell into that group just based on the data, which is an interesting result for us. Next slide, please. So one of the outputs of the MB Abund analysis was that we got which of the flow metrics, if you remember, we had 8 at the beginning, were influencing the community structure as a whole. So we get a community level response, and we see that 6 of the flow metrics were significantly affecting the community. Next slide. However, what's probably more interesting is how individual flow metrics are impacting individual groups. So we have the different groups from the dendrogram that we showed just a minute ago, and different flow metrics are affecting or significantly related to the response of that particular group. And I'm just going to show a couple of quick examples from some of the outputs. So if I could have the next slide, please. This is the response of the Minnow group. They have a very similar, most of them, reproductive strategy. And they were related to 2 of the flow metrics. They were reduced at more variable flows, but they increased with flood frequencies. And this group is interesting because it included the red shiner, which was ubiquitous across many of our samples and actually made up a large proportion of the community. And it's suggesting that, you know, even a reduction in flood frequencies affects species that are very common. The next slide, please. Then moving on to kind of the Pelagic Broadcast spawning group, we saw that there was a kind of drastic reduction in the relative percentage of this group as the minimum flow date moved through time. And what we attribute this to is a relationship between minimum flows and also when the species is spawning. So this species is thought to spawn during high flood pulses. It also needs a certain flow for its eggs and ichthyoplankton to drift downstream. And if you have very low flows during the spawning period, it's likely that the eggs or ichthyoplankton can drop out of suspension and then maybe more vulnerable to abrasion or unsuitable conditions or predation. And this relationship with kind of the low flows has been, and this group of particular species has been seen by some work from Texas Tech University and Gene Wild's group there, who related reproductive success to the requirement of actual flows. Next slide, please. So just a quick bit in summary. Baxter's group modeled discharge for a large area of the Canadian River, and we translated that into ecological flow metrics alongside Jonathan Kennan, who's at the New Jersey Water Center. We've split the community up into a number of trait groups, which combines both the morphology but also some of the reproductive strategies. And we've linked that to specific parts of the flow regime, which is kind of interesting in terms of ubiquitous species, like I said, like the red shiner, were still being influenced by high flows, and some of the threatened species, such as the pelagic broadcast faunas, were impacted by a different part of the flow regime. Next slide, please. We just have a few more bits to do in terms of that. In terms of this is a spatial model. We're going to have a look at the non-linear relationships and try and maybe strengthen some of the modeling. There's a potential issue with zero inflation. We have lots of zeros for some of the trait groups. And also compare that with maybe another method in maybe something like boosted regression trees. And then kind of adding in the climatic scenarios that Baxter highlighted earlier, and this links onto the decision support tool, which I'll talk about in a second, but seeing how the community responds to these changes in flow mediated by changes in PET and precipitation. Another aspect of the project is to look at the trends in the community, not at this whole suite of 75 sites, but at a subset, about 11 sites, where we have longer community data. Next slide. And just to wrap this up, as highlighted by both myself and Bill, we're hoping to kind of build together kind of a web-based interface where people can go and have a look and sort of interact more with the results of this study. Hopefully this will make it more kind of accessible to the funding bodies, interested parties, but also the wider public. The general idea is to sort of visualize the response of the community to the different climatic scenarios that Baxter's provided us, and allow people to change the scenarios and see how that impacts the community. There is online at the moment at that web address another decision support kind of tool related to some work that we did on the Arkansas River Shiner. It's very much at the sort of first phase, so not all of the links work, but if anyone's interested, go and have a play with that, and it will kind of give a background of what we're kind of thinking in terms of the decision support tool. Okay, and that's all from me. Thank you very much.
Okay, thank you very much, Tom. Really appreciate you calling all the way from Wales and giving up some of your dinner time over there. That's the last slide that we had. If there are questions from the attendees, we'd be happy to try to answer them.
Anyone who has a question can press star on their phone to unmute.
All right. If we can you hear me, Blair?
Yes.
Yes, I can.
Yeah. Thanks again, Bill and Tom and Baxter. Great presentation, and maybe you might get some follow-up questions. And for folks on the phone, if there are others in your organization who would be interested, we will post the recording of this webinar on the GCP website as well. That should be up in the next couple days. So I don't see anyone has raised their hand or anything, so maybe we'll, if there's no pressing questions, we'll sign off for today. And thanks again very much Bill for the presentation. And just a reminder, our next webinar will be on Wednesday, October 7th. And that day we'll be talking about some of the land cover work we've been doing in Oklahoma with the Oklahoma Ecological Systems project. So thanks very much, everyone, and thanks for joining us today.
Okay, thank you.
Thank you.
Thank you. Bye-bye.